or other than chlorophyll pigments. This chlorophyll pigment is a material which helps in performing photosynthesis. And that material is stored in a plate-like structure, which is called as thylakoid. And those thylakoids are arranged in stacks in chloroplast. So that much important structure or that much important organelle is chloroplast. Now look at the structure of a chloroplast and its history. Usually photosynthesis takes place in green plants. That means in green parts of the plant, like leaves, stem, and floral parts. So in all the green color appearing parts, we can see the chloroplast. And this chloroplast first structure, it was observed by N. Grew in 1682. But later on, Schimper explained the structure of chloroplast. So discovered by Grew, and the detailed structure is given by Schimper. And this chloroplasts, they are arranged parallel to the mesophyll tissue. We know the structure of leaf, upper and lower epidermis, and in between these two layers, whatever tissue is there, that is treated as mesophyll tissue. And this mesophyll tissue, that means parallel to the mesophyll tissue. That means in this mesophyll tissue, all the chloroplasts are scattered and they are parallel to the cells. And this helps. Why they are parallel to the mesophyll cells? That means this arrangement helps in diffusion of CO2 into chloroplast. That is convenient for the diffusion of carbon dioxide from the upper cells, upper epidermal cells into the mesophyll cells. So because of that reason, all the chloroplasts are arranged parallel to the mesophyll cells. The next, chloroplast. If we observe the structure of chloroplast, it is surrounded by two membranes. Look at the diagram here, outer membrane and inner membrane. So each and every chloroplast is bounded by two membranes, outer layer and inner layer. In between these two membranes, yesterday we have seen in this diagram. Yes, here outer membrane and inner membrane. Between these two membranes, there is a gap. There is a space which is called as intermembrane space. And here is also, it is there. So this gap is treated as intermembrane space. Now, inside these two membranes, there is a protein colloidal complex. And this complex is called as stroma or matrix. And this matrix that contains various enzymes and DNA and RNA and ribosomes. And next, this structural and functional unit of chloroplasts are thylakoids. That means they are the chiefest part of a chloroplast. And this thylakoids, they are treated as structural and functional unit of this chloroplast. Like in living organisms, the cells are the structural and functional units of that organisms. In the same way, for the structure and for the structure of the chloroplast and for the function of the chloroplast, it badly requires this part which is called as thylakoids. That's why it is called as structural and functional unit of chloroplast. And these thylakoids are embedded in the matrix. That means they are arranged in the stroma or in the matrix. That means they are sunken or they are embedded in the stroma part of the chloroplast. And if we observe them keenly, that thylakoids, they are arranged in groups. Look at this diagram. Here it is. So these thylakoids are arranged in groups. And this group of thylakoids are considered as grana. So what is grana? Grana is a group of thylakoids. The thylakoids are plate-like structures. They are inserted in one another. That means that the structure of each and every thylakoid is similar to one another. That's why if they are in the same size, shape, or structure, then easily they are inserted in one another. That's why they are appearing in groups. So these are all thylakoid plate groups, and name for this group is grana. And in each group we can find a 40 to 100 this groups, nearly how many groups are visible in a single chloroplast? Means 40 to 100 grana groups are present or visible in this stroma or matrix. That means granule groups are nearly 40 to 100 in each chloroplast. And in each grana, how many thylakoids are able to present? Means in each grana, nearly they are 50 to 60. 50 to 60 thylakoids are present or arranged in each group of grana. Then, if we observe the structure of thylakoid, each thylakoid is a flattened sac-like structure or plate-like structure. It's a plate-like structure. Then, in this plate, usually we use as a plate for putting something or arranging food stuff in our daily life. In the same way, this plant, it uses this thylakoid plate-like structure for keeping the pigment or arranging the pigment in this plate-like structure. So thylakoid is a plate-like structure in which this pigment material is arranged inside the plate. So this is about the thylakoid. Thylakoid is a plate-like structure in which we can find the green color chlorophyll pigment. Then we are observing these granule groups, nearly 40 to 100 in this chloroplast. And these granule groups, they are connected with one another. Look at the diagram here. These granule groups are interconnected. They are connected with one another by means of some strand-like structures, some fiber-like structures. And these strand-like structures are called as stroma thylakoids. Granule groups are interconnected by means of fiber-like structures, which are called as stroma thylakoid. So this is about the structure of a chloroplast. Once again, this chloroplasts are chloroplasts are the organelles which helps in storing green color pigments in their structural functional unit thylakoid. And this chloroplasts are first of all observed by N. Grieve in 1682, and a detailed structure was given by Schimper. And if we observe the position of chloroplasts, that means where the chloroplasts are arranged and how they are arranged. Chloroplasts are observed in all the green parts of the plants like leaves, stem, and other floral parts. And if we observe the chloroplasts, their position, and they are present in mesophyll cells of the leaf, and also in mesophyll also they are chloroplasts are arranged parallel to the mesophyll cells. Why? Reason for that type of arrangement is to absorb the CO2 or for CO2 diffusion, for easy diffusion of CO2 into mesophyll cells. This chloroplasts are arranged parallel to the mesophyll cells. That is the reason for that. And each chloroplast, if we observe the structure of chloroplast, each chloroplast is bounded by two membranes, outer membrane and inner membrane. In between these two, there is a gap, which is called as intermembrane space. Inside the structure or inside these membranes, we can see the presence of gelatinous stroma or matrix, in which different types of enzymes and the DNA, RNA, and ribosomes are present. And in this stroma only, we can see the arrangement of so many groups like structures, which are called as grana. And this grana is nothing but a stack of thylakoid structures. What is thylakoid? This thylakoid is a plate-like structure, and the number of thylakoids are arranged in groups, and that group is called as grana. And these granas are interconnected by means of thread-like, fiber-like structures, which are called as stroma thylakoid. And this thylakoid is helpful in storing the pigments. That is the structure of chloroplast. Next slide. Yes. John Ingenhousz, one more scientist. He made his experiments in proving that sunlight is essential to the plants to perform photosynthesis. Yesterday, we discussed the factors of photosynthesis. There are 4 important factors. Out of 4, one of the factors is sunlight, and that sunlight is essential for to perform photosynthesis. And this fact was proved by John Ingenhousz. He belongs to the period 1730 to 1799, and he proved that sunlight is essential to the plants. And by conducting one experiment, which is called as Hydrilla experiment, he proved one more thing, that he stated that green parts releases oxygen during the photosynthesis. Yes, to prove this, that means plants performs photosynthesis and they releases oxygen during the photosynthesis. That is proved by performing a experiment which is called as Hydrilla experiment. And for that he took a glass jar or beaker and one test tube and one thistle funnel and a few Hydrilla plant branches or twigs. And this Hydrilla is an aquatic plant, and the rate of photosynthesis very rich or very speed. That's why to observe it very soon, they selected this Hydrilla experiment for their experiment. And the arrangement of this experiment is, first of all, this beaker is filled with water 3/4. 3/4 of the beaker is filled with the water. Then he took the thistle funnel, and from the broader end, he arranged the twigs, Hydrilla twigs, into its stack or stalk-like structure. So these branches are arranged through this broader end of the funnel into the stalk of the narrow end of the funnel. Like this, he arranged the Hydrilla experiments. Then He took one test tube which is filled with water also. So this water-filled test tube is arranged invertedly on the stack of the thistle funnel. He arranged the water-filled test tube on the stack of the funnel, or test tube is inverted on the stack of the funnel. Like this, this arrangement is kept in the water or at the bottom of the flask or this glass beaker. Funnel's broad end touches the bottom of the beaker. In this way, arrangement will be made, and this experiment will be kept in under the sunlight, or it is arranged outside of the open area where the sunlight will be available. Now this arrangement is left for 2 to 3 hours, and after 2-3 hours they observe the experiment. Any changes? Yes, they observe the bubbles coming out from the stack of the thistle funnel into the test tube, and a decrease of water. Initially, before arranging it in the open area, they marked the level of the water in the test tube and also level of the water in the beaker. So both the levels are marked before arranging it in under the sunlight. And after 2-3 hours, they again observe the water levels in test tube and in beaker. In test tube, water level is decreased, that is coming down. That was observed, and water level in the beaker gradually increased. That change was observed after 2 to 3 hours time. So there is a change in this experiment. There is a level of water decreased in test tube and level of water increased in the beaker. And moreover, from the stack of the thistle funnel, bubbles are coming out into the test tube. So these are the changes observed during this experiment. And the bubbles release the small bubbles and they, yes, these bubbles, how they are coming out means what is a gas inside that bubbles was tested. And now this Hydrilla twigs, they are getting all the factors. Here water is available to the twigs, sunlight is available to the twigs, and carbon dioxide, then chlorophyll, all the factors are there. So it performs the photosynthesis. As a result of yesterday, we have observed the equation of the photosynthesis. See here. So end products are glucose, oxygen, and water. So here also in Hydrilla experiment, this Hydrilla twigs performs photosynthesis and releases oxygen, and that oxygen which is released, that comes out on the top of the this test tube. Why? Because this oxygen air is lighter than the water. That's why it reaches the top of the test tube where the empty place is there. So in the form of bubbles, that air enters on the top of the test tube, and that gaseous oxygen shows pressure on the surface of the water to occupy it, to occupy the place. These bubbles, they shows oxygen which is in bubbles shows pressure on the top of the water surface in the test tube. As a result, that water comes down into the beaker. As a result of that, water level in the beaker gradually increases. So by this, we can say this oxygen is released into the test tube by this Hydrilla twigs as a result of photosynthesis. And this was also be proved by putting a matchstick into this test tube that continues to light. That is the proving of oxygen. So in this way, John Ingenhousz explained that photosynthesis process results in the formation of glucose and water along with those oxygen also. So this was by John Ingenhousz. Next. In our previous slides, we have discussed that the chloroplast is the most essential organelle of the plant, which helps in performing the photosynthesis. So that chloroplast is helping in storing the pigment like a chlorophyll. So this chlorophyll is a main important material to perform photosynthesis. So we have to know the structure of chlorophyll because it is an important pigment to perform photosynthesis. That's why we should know the structure of chlorophyll. Here is the chlorophyll molecule and its structure. It is looking like a head with a tail, isn't it? So this head portion is made by what, and this tail portion is by what? We should know the details. Here, chlorophyll A. In chlorophyll also there are so many varieties. Chlorophyll A, B, C, D, E, like that. And there are 7 types of chlorophylls, and other than this chlorophyll, other pigments are also there. Now we are going to discuss chlorophyll A. This chlorophyll A contains centrally located magnesium. See here, centrally located magnesium ion, and it is encased in a large ring structure which is known as chlorine. That means this is the magnesium, and there's a blue color structures are nitrogen molecules, and this nitrogen molecules makes a round structure. So magnesium ion encased in a large ring structure known as chlorine, and this chlorine ring is a heterocyclic compound derived from pyrrole. Pyrrole structure. That means 4 nitrogen atoms from the chlorine surrounds and binds the magnesium atom. That means here this green color is magnesium and surrounded by 4 nitrogen molecules. So this each group is called nitrogen group, makes one pyrrole, and this magnesium centrally located magnesium uniquely defines the structure as a chlorophyll molecule, and this tail-like structure is called as phytol. So in chlorophyll structure there is a round head-like structure and a tail-like structure. This head is made by one magnesium molecule and which is surrounded by 4 nitrogen molecules, and each nitrogen makes one pyrrole structure, and at one end we can find a tail-like structure which is called as phytol. This is the structure of chlorophyll. Then next, there are just now only said that chlorophyll pigment. In chlorophyll pigment, we have so many types of chlorophyll pigments, and about that now we'll discuss. In higher plants, there are 2 types of chlorophyll pigments. Usually in not in all, in higher plants there are 2 types of chlorophyll pigments. Number 1 is chlorophyll A, and number 2 is chlorophyll B. Chlorophyll A and chlorophyll B are the main important types of pigments in chlorophyll. They both are similar in their molecular structure, but exceptionals are there. There is a difference between those 2, chlorophyll A and B. What is that difference means? CH3 group is in chlorophyll A. That means methane group is attached to one end of the chlorophyll A. And the CHO, aldehyde group, is attached in chlorophyll B. That is only the difference between A and B. In chlorophyll A, which group is connected with the chlorophyll A molecule? Means methane group is attached or is connected to the chlorophyll A molecule, whereas CHO, aldehyde group, is connected with chlorophyll B. Now, chlorophyll A is important in absorbing light energy and converting it into chemical energy. This in both of these, chlorophyll A and B, chlorophyll A has much more importance. Why? Because during the photosynthesis, it helps in absorbing the light energy and converting it into chemical energy. That's why this chlorophyll A is most important pigment. Then next, there is a difference. Differences between chlorophyll A and B as given in a tabular form. What is a chlorophyll A molecular formula? Means C55H72O5N4Mg is its molecular formula. Whereas chlorophyll B, C55H70O6N4Mg is a chlorophyll B molecular formula. That means centrally located magnesium, which is surrounded by 4 nitrogen molecules, and other these molecules are surrounds these pyrrole groups, right? And here in chlorophyll A, carbon 3 contains CH3 group. Just only we have discussed that the difference between chlorophyll A and B. CH3 methane group is attached to the chlorophyll A molecular and cheptunnamu. That means in chlorophyll A, carbon 3 carbon C55 is there. Carbon number is C55. In out of 55, third carbon, it is connected with the methane group. That is third carbon. Carbon number 3 contains a methane group. That's why it is chlorophyll A. Whereas in carbon 3 only, position is same, but groups are different. For third carbon, here CH3 is connected, and here for carbon number 3 only, it is connected with CH3 group. That's why CH3 group to the third carbon indicates chlorophyll A, and a CHO group, that means aldehyde group, to the carbon number 3 indicates chlorophyll B. The next difference between chlorophyll A and B is chlorophyll A is soluble in petroleum ether. It is soluble in petroleum ether, whereas this chlorophyll B is soluble in 92% methyl alcohol. It is soluble in methyl alcohol, not in petroleum ether. The next difference between these two is chlorophyll A helps in absorbing red color wavelength light, and here chlorophyll B, it helps in absorbing violet and blue color wavelengths. We know the wavelengths of this colors, light, red color, that is of what type of wavelength it is having, and violet and blue also. It is different with the different wavelengths. So these are the important differences between chlorophyll A and B. Once again, chlorophyll A, its molecular formula is C55H72O5N4Mg, whereas chlorophyll B, C55H70O6 and N4Mg. And main important difference between these two is methyl group, methane group is connected with C3 carbon, And here aldehyde group is connected with C3 carbon in chlorophyll B. And chlorophyll A is soluble in petroleum ether, whereas chlorophyll B in 92% methyl alcohol. And in Vibgia, red color wavelength is absorbed by chlorophyll A, and the blue or violet color wavelength light is absorbed by chlorophyll B. These are the important differences between chlorophyll A and B and types of chlorophyll pigments. The next, go for the other slide. Yes, this is accessory pigments. What is the accessory? Means the main role is played by chlorophyll pigment in photosynthesis. In addition to this chlorophyll pigment, there are some other pigments around this chlorophyll, and those other pigments molecules or pigment molecules are treated as accessory pigments. That means other than chlorophyll A and B pigments, there are some other pigments which helps in transferring the photons from them to the chlorophyll A or B. Then those pigments are called as accessory pigments. Here it is a definition. See, pigments which are involved in the process of photosynthesis in addition to chlorophyll A are called as accessory pigments. Yes, this is the right definition. What is that? In addition to chlorophyll A, whatever other pigments are involved in this photosynthesis process are called as accessory pigments. And what are they? What are the other pigments which comes under accessory pigments? Means one is carotenoids, the other one is phycobilins. Carotenoids and the phycobilins togetherly called as accessory pigments. Then first of all, look at the carotenoids. What are these? And what color they are? See, carotenoids are a red, orange, yellow color pigments which absorbs blue and violet light or blue and violet wavelength light. So carotenoids are especially they are in red, orange, yellow color pigments. Red, orange, yellow color appearing different examples are there. For example, neem fruit, lemon fruit is yellow in color, orange tomatoes are there, red color leaves are there. So all those parts which are appearing in different yellow, red, orange color are because of having this carotenoids. And this carotenoid pigments, they especially absorbs blue and violet color wavelength light. And this carotenoids are also embedded in the thylakoid of chloroplast. That means along with the chlorophyll pigments, these carotenoids are also embedded in the thylakoid. And these pigments protect chlorophyll from photooxidation. Yes. So from protecting them, that means protecting the chlorophyll pigment from photooxidation. These accessory pigments like carotenoids are useful. And it is an accessory pigment act as an antenna complex in absorbing light and sending them to the reaction center and converting them to the chemical energy. So here, this carotenoids, 1 minute. This carotenoids, they act as a complex. They act as a complex. That means centrally chlorophyll pigment is situated around to the chlorophyll pigment. This accessory pigments like carotenoids are arranged, and this accessory pigments they helps in absorbing the solar energy in the form of photons and transferring the photons to the reaction center or transferring the photons to the chlorophyll A pigment and converting the solar energy into chemical energy. So this is the role of accessory pigment. So what is the role of accessory pigment? The role of accessory pigment is to transfer the photons to the reaction center. Usually reaction center is chlorophyll A majorly. So minorly chlorophyll B. So majorly chlorophyll A receives additional photons from the accessory pigments. So they act as antenna complex in absorbing the solar energy and in the form of photons and transferring them to the reaction center is the main job of carotenoids or accessory pigments. The next step, these carotenoids are divided into two types. One is carotenes and the other one is xanthophylls. Carotenes, usually in what color they are, in which parts we can observe them, means carotenes are usually orange, red color pigments. Their molecular formula is C40H56. And they are orange and red color pigments. Like example is carrot. That carrot appears in red color, or tomatoes, they are red in color. So orange or red color. Carrot is nothing but orange in color. The tomatoes are red in color. So in that parts, we can observe the carotenes. And the next one is xanthophylls. Xanthophylls are purely yellow color pigments. You know the yellow color different structures. Just now only told you lemon fruit, which is yellow in color, and leaves. You know, after crossing their age limit, different parts they turns into yellow. So because of the development of xanthophylls, they turns into different, green to yellow or red to yellow, like that. So in all the yellow appearing parts, we can see this xanthophyll pigment. And in this xanthophylls, one important pigment is there, that is fucoxanthin. It is an important xanthophyll, and it is especially appears in Phaeophyceae family members. So this is about the accessory pigments in which carotenoids shows these characters. So carotenoids are red, orange, and yellow color giving pigment, and it absorbs blue and violet light from the WebGR, and these carotenoids are also present. Carotenoids are present along with the chlorophyll pigment in thylakoids. So that means these carotenoids are embedded in the thylakoid. Right. Then this carotenoids helps in protecting the chlorophyll pigment from photooxidation, and this carotenoids they act as antenna complex in absorbing the solar energy in the form of photons and transferring it to the reaction center and changing it from solar energy to chemical energy. And these carotenes are of two types. Carotenoids are of two types. One is carotenes, other one is xanthophylls. Carotenes, usually they are red or orange in color. Or this carotenes having parts appears in red or orange color. Example is carrot, and its molecular formula is C40H56. And xanthophylls, usually they are yellow color giving pigments. This yellow color appearing parts are structures, all they contains this xanthophylls. And in xanthophylls, picoxanthin is an important pigment, which is especially found in phaeophysic members. This is about the carotenoids. The next, another accessory pigment is phycobilin. What is phycobilin? Means it is a red, green algal members. It is available, and these phycobilins are of three types. One is phycocyanin, other is allophycocyanin, and next one is phycoerythrin. Phycocyanin and allophycocyanin both are blue color giving pigments, and phycoerythrin, it is red in color. For example, red algae, red algal member, rhodophyceae algal members, they consist or they have phycoerythrin pigment. And this blue-green algae, it consists phycocyanin. So phycocyanin and phycoerythrin togetherly treated as phycobilins. Both of these pigments are togetherly called as phycobilins. And this phycobilins helps in trapping the sunlight and transporting of the sunlight to the chlorophyll pigment. That's why all the pigments, that means chlorophyll pigment or accessory pigments, if we observe them keenly, one thing we can understand that these pigments helps in trapping the sunlight or trapping the sunlight in the form of photons and transporting it to the reaction center. That is the main important role of this pigments. And next is quantosome. What is quantosome? See here, each chloroplast consists of 50 to 60 quantosomes. Granule membrane is made with proteins and phospholipids, and Park and Pun 1961 reported that the presence of a dome-shaped granular bodies inside the granular lamellae, and Park and Begins coined the term quantosomes for granular structures, and these are considered as basic unit of photosynthesis. During photosynthesis, 2,500 chlorophyll molecules are required to fix one molecule of carbon dioxide, which is equal to 10 quantosomes. So what is a quantosome means? It's a fundamental unit. It is a fundamental unit of granular lamellae. So quantosomes, these are considered as basic unit of photosynthesis. So that is about the quantosomes. The next. This is the equation of photosynthesis. So with this, we have completed the introduction portion of the photosynthesis. That means where the photosynthesis takes place and what are the structures involved in this and what are the investigations done by different scientists to prove the factors are essential to perform photosynthesis. All these are discussed in our previous slides. So these are the factors. Factors are of four types: sunlight, water, carbon dioxide, and pigments. These four are the raw materials or factors which are essential to perform photosynthesis, and each one is proved by different scientists that the factor is essential to perform this photosynthesis. First one is Mole's half-leaf experiment, in which a leaf is tested by inserting half of the leaf into the bottle and half of remain with outside of the bottle. And in this experiment, half of the leaf which is inside is able to synthesize, is not able to synthesize of starch because of the lack of carbon dioxide, which is absorbed by potassium hydroxide crystals. And outside of the leaf, it is getting all the factors, all the 4 factors. That's why it performs photosynthesis and turns into blue color by iodine test. And that experiment is called as Moll's half-leaf experiment, which proves that CO2 is necessary for photosynthesis. This was explained in our previous class. The next, during this Priestley experiment, in Priestley experiment also we have proved that air, Priestley proved what he concluded, that plants helps in regenerating or restoring the air in the atmosphere, was concluded by Priestley by performing a series of experiments in which 2 living organisms which avails oxygen for their life, and after some duration, both of the organism, that means candle was lit up and the rat was dead. But by arranging one potted plant in another experiment, along with these 2 living organisms, Priestley arranged one potted plant and under the same bell jar. After some time, 3 of the organisms are in the same positions, which they are continuing their lives safely. By this, what we can understand is whatever oxygen is availed by both of these organisms, that means rat and candle, will be regenerated by the plant by the photosynthesis. That means oxygen is released, carbon dioxide is absorbed. That means carbon dioxide is taken in and O2 releases out into the bell jar. As a result of this, these 3 are safe. So by this, we can say that plants releases oxygen and carbon dioxide is received by the plants. And this experiment was explained by Priestley, and the plants helps in restoring the restore to the air. Whatever air is consumed by breathing animals is again regenerated by the plants. Was explained by Priestley. And Engelmann, he discovered the effect of different wavelengths of light by putting a prism and passing the light through this. That splits into 7 different wavelengths of colors, which is called as Vigia. And in this, red and blue color wavelengths are useful. Red and blue color wavelengths are useful for the photosynthesis. And Cornelius van Niel, he stated that O2 is evolved by green plants, not from O2 is not from CO2, and this O2 is released from H2O, not from CO2, and this was proved by Van Niel. Then Sachs, he discovered, noticed that the starch is the end product of photosynthesis during the light phase or light focused chloroplast. That means chloroplasts are essential structures which receives the light energy, and they continues photosynthesis and resulting starch as the end product, and that was by Sachs. And for chloroplast, structure of chloroplast, important structures are grana, and in grana, structural and functional unit of this chloroplast is thylakoid, in which pigments are stored, are preserved, and in this only the light energy will be trapped and converted into chemical energy. Then Ingenhousz proved that oxygen is released during this photosynthesis by performing one experiment which is called as Hydrilla experiment. The next, by all these slides, we can understand that the photosynthesis is mainly dependent on this chlorophyll pigment only. So we should know the structure of chlorophyll for this understanding sake. Chlorophyll, it shows spiral structure, four groups which are linked with the magnesium molecule in the center, magnesium in the center, and surrounded by nitrogen molecules, four nitrogen molecules, which makes four groups of pyrroles, and